Alright, welcome everyone to our second lecture. So today's lecture is going to be on DNA replication, the general mechanism of DNA replication. So I'm going to explain what it means, the process, and the sort of like some differences between the process in eukaryotic organisms and prokaryotic organisms. Alright, so when we talk about DNA replication, you need to understand what led to that kind of like suggestion that the DNA molecule replicates. Now, we know that before growth can occur, there has to be division of cells, because the division of cells leads to growth occurring. And then another thing that we also need to know is that when cells divide for prokaryotic organisms during the process of binary fission and eukaryotic organisms, mitosis. So when cells divide, we need to know that the genetic instruction is required for cells to survive, required for life processes to occur. So if a cell divides, definitely the cell has to find a means to transfer its genetic material from one generation to the next generation. So that suggested that the molecule of life had to be replicated. But scientists for a long time, during the early period of this DNA era, they were trying to find out how this occurred. So they suggested three models. The first model is the conservative model. The second model suggested was the semi-conservative model. Then the third, the dispersive model. So these were three models of DNA replication that were suggested. Now, the conservative model, what it just says is that during the process of replication, take for example, if this were to be the DNA molecule, let's say there's a process of replication, then what you notice is that one of the daughter molecules, this is the parent molecule, one of the daughter molecule has is exactly the same as the parent molecule. And the second daughter molecule is a copy of this daughter molecule. Well, looking from the lenses of our present knowledge, this looks kind of like absurd. Why? Because it wouldn't be energetically favorable. Based on the double molecule structure, the sugar phosphate is found outside, and then the base is found inside. It won't be energetically favorable. To mean the DNA will have to break the hydrogen bond, then after synthesizing it, after popping it, probably have to break again. So then another thing is that it will be error prone. So the next model is this. So take for example, this is the parent molecule. Then during the process of replication, you have in the two daughter molecules, one of the strands comes from the parent molecule, while the second strand is a new strand, newly synthesized strand. We'll come back to that model later. And let's look at the dispersive. So this is the parent molecule, double-headed structure. Don't forget. Then you have dispersive, something like this. You have something like this. It's quite complex to draw, but just try my best. Let's see how to do. Okay. So the dispersive was simply saying that in the daughter molecule, we have a random or dispersed distribution of the parent molecule within the daughter molecules. That's what they're talking about. But you will remember from our previous video that the DNA was discovered by who? DNA structure was discovered by who? Watson and Crick. So based on the Watson and Crick model, they also suggested that there was a form of complementarity between the bases. So based on that model, Watson and Crick adhered to the semi-conservative model of replication. So due to some experiments by certain scientists, they were able to confirm this. This has not been confirmed, doesn't mean that it will not come. But this is the model of DNA replication. Alright, so let's look at the process. Let's look at the process. DNA replication. Now, you remember from our previous video, they explained that DNA replication occurs from the 5' to the 3', and then the DNA is antiparallel. So I'm just linearizing the DNA, so it's actually double benefit for us to understand and to observe it. So what we are saying in essence is that replication occurs, and then from one you have this 5' to 3', this is 3' to 5', and you have this 3', yes, 5', and this is going to go 5' to 3'. You see this? The red represents the new strands that are missing the side. Alright, but let's look at the three processes, the three steps. There are three stages of DNA replication. Now mind you, DNA replication in eukaryotes is not exactly the same as that in prokaryotes. However, they are very similar. So you check some textbooks, you may be confused because you see some very absurd names. You need to know that the typical general mechanism of DNA replication is what in the textbook is from that of Escherichia coli. So, but that in eukaryotes still requires a lot of research and everything, but some work has been done on it by studying yeast as a model. So the best model for DNA replication is the Escherichia coli. However, it may slightly differ the structure of different proteins. Some proteins may require cofactors and other things, but let's just use this general mechanism. Alright, so E. coli, for example, which is the general mechanism. The first step is initiation. Initiation. Now, what do we mean by initiation? The first thing in initiation is this, that we need to find something that is called an origin of replication. Origin of replication. Now, the origin of replication is known to have a high AT-rich sequence. AT-rich sequence. Now, there's a protein, the initiator protein, helps to find this origin of replication, and the origin of replication in Escherichia coli is known as ORIC-C. I think this means origin of chromosomal replication. Let's go. It's ORIC-C, O-R-I-C. And then there is a particular initiator protein that recognizes it. That protein is DNA-A. DNA-A. So, take for example, DNA-A comes here and binds to this region. It leads to the formation of what we call a replication bubble. Replication bubble. So it's a bubble, it looks like a bubble, replication bubble, due to the binding of the... And you remember from the previous video that 80, the number of hydrogen bonds between adenine and thymine is actually how many? 2. And between guanine and cytosine is actually 3. So it's easier for you to separate the bonds between an A-series sequence and that of a G-series sequence. So that's why it's easier. So it will lead to probably phosphorylation or something will change the conformation of the protein, which will therefore lead to the breakage, weakening of those bonds and the separation, and thus the formation of the replication bubble. Now, the next thing we notice, there is a protein called helicase. Helicase. Helicase is DNA B in Escherichia coli, and there is a certain protein that helps to load the helicase. It's called the helicase loading protein. Helicase loading protein is also known as DNAC. DNAC. So imagine helicase comes here. Alright, helicase coming here, and probably you have the helicase loading protein somewhere here. So helicase is loaded onto the DNA, and then I can send it the 5' to the 3' region, the 3' to the 5' region. Do you get this? Okay. So then this is not all. The next thing is that there is a protein called primase. Prime is also known as DNAG. DNAG.